So there's a bit of a crisis on planet Earth, as we're all aware, and probably a lot of you here are also aware that there are some solutions, quite a few solutions available to us. When we're talking about these transgressions of these boundaries, biosphere integrity seems to be a pretty serious one, and particularly biodiversity loss. And agriculture has a huge role to play in not only causing this issue, but also helping to solve it. So it's quite clear, and again, I'm sure this has been motivated already, but just to remind you that when we diversify our agricultural systems, we not only benefit natural biodiversity in general, but we also can pick up a lot of ecosystem services which have positive effects. And ultimately, this can be done if we design our systems well without losing the performance, the agronomic performance of the system. So ultimately, the yield per unit of input that we're putting into it. Now, as Joachim said, it was actually Joachim and Liesje that came to the plant breeding group a few years ago and inspired me on this track, I suppose. I mean, I was doing this sort of stuff to some extent before I came to Wageningen even. But yeah, so Joachim came and he asked us, is anybody in the plant breeding group thinking about breeding for intercropping? And the first question, I guess, that arose to me, primarily coming because I used to work in a little gene bank in Ireland, looking after old varieties of vegetables and grains and apple trees, was, are these modern varieties that we have now adapted? These new high input, high response varieties, gene types, are they well adapted for these more diverse systems? If they're not very well adapted, how should we go about breeding then for these systems? So that's pretty much the focus of my presentation. This is a very slow, it's not, I don't think it's a battery, it's just a very slow response. It's because it's going through here and here and here and here. So I'm afraid I'm going to go back to the old school of just pressing the button because I've got very little patience. We're
not in a rush.
No, okay, but I've had 2 coffees, so my patience is really low. So myself and Joachim and a few others actually wrote did a small review about this, about breeding beyond monoculture. If we're thinking about moving away from monocultures, how can we breed beyond that? But a couple of months later, actually, Virginia Moore and some colleagues in Cornell also wrote a review along similar lines, and actually thought this review would be a nice way to structure today's talk, primarily because she had a lovely colorful decision tree, or sort of a decision wheel. And of course, this breaks it down. I mean, there was a question about how do farmers make decisions, and you said, well, maybe we can use models to help them. But of course, maybe not all farmers have access to these high-tech models. This is the sort of a decision tree for a breeder. So how does a breeder approach this? And I thought it was quite nice, but I also thought it wasn't entirely right. It's somewhat flawed. And of course, well, we wrote the other review just a few months, so it's of course it's okay for me to I think this poses some nice questions. So what I'm going to do is I'm going to use this as a bit of a framework for today's talk. Now I'm going to focus first of all on this first quadrant, which is, well, do we actually need to breed for it? And without going through all of these points in detail, the first question that Virginia and her colleagues raise is actually, we've got potentially constraints in our system. Over there,
because of
the light.
Yeah.
You prefer me over here?
Yes.
Okay, this is my good side. So.
Yeah.
Okay. So can we address the constraints of the system more effectively using an intervention, using management, maybe playing around with density, fertility, timing of sowing, those kind of things, rather than just straight going towards breeding? I mean, this is of course you have to know your system. A breeder wants to know the system before they start to breed for it. Of course, I don't think, I mean, you'll see with these things, it's a yes, no, it's a decision tree. So if yes and if no, so it's presented as if it's a very simple yes, no question. But I would argue that this is probably not trivial at all. We were talking about these statistical interactions, but your genotypes could quite likely interact with density, or as we just saw, for instance, the fertility regimes and so on. So it's not just so simple to say, well, we'll play around with the density and we'll play around with fertility or some other intervention and manage it, if the genotype that you choose has a big influence on the outcome of that intervention. So this is not trivial. It's already a tricky problem, and we're just at the first point. The second point that I'm going to talk about a little bit is assessing variation. So do we have sufficient variation within the component species? Do these component species show any variation for performance? And again, it's a yes, no type answer here. But again, I think somehow this answer, if no, the conclusion, don't bother with breeding. Maybe we're a little bit fast, or as the Dutch say, kort door de bocht, to just say no, therefore don't breed. Because oftentimes, if a breeder comes up with this sort of a no, they'll go a step further. They'll look a bit maybe wider. You know, they'll test beyond the set of varieties or cultivars that they're screening. Maybe they'll introduce novel variation. I mean, there's new legislation regarding gene editing. There could be ways to get around this constraint if we don't have enough variation already within the species that we have. Mm-hmm. On to the second quadrant for designing a breeding scheme. I'm going to go through this in a little bit more detail. And the first, Moore et al. Suggests that we should use a genotype type management or genotype times management interaction as a sort of a flag or an indicator to tell us something about, well, about whether we need to breed or not. I'm going to look at that in a bit more detail. Is G times M the most appropriate way to do it? Is there some other measure that we could consider? Then Moore talks about these traits of interest. And again, this paper was sent around to you. So for those of you who have done your homework, you've already seen this decision tree. This was the one that I offered. So apologies if I'm going a bit quickly. But
Peter, the management here is the choice intercrop or monoculture, or other types of management as well.
In this instance, you could consider management as monoculture versus intercropping. Yeah, but of course, there's so many different types of intercropping. The management's become it's not just a two-level treatment, shall we say? So are these key traits observable in monoculture? And of course, this is an extremely pertinent question. If they are observable in monoculture, then why would you bother setting up all these mixture experiments? You know your traits, a bit like Bourke, as he's been suggesting. You know your traits. You can select for them. You can make your perfect ideotype of your maize plant or your faba. And this is the one then that will work in a particular intercropping setting. But again, I don't think it's as simple as that. First of all, we don't know what these key traits necessarily are. And I think this is the point that Maduri was making very nicely. And there's also the question about plasticity. So these traits may vary in how they express themselves depending on the cropping partner, the density, etc., the soil type. So trait plasticity is very important. So it's not just that a trait is some sort of a fixed concrete stone thing that we know about and we've got to capture it. These traits are always varying and different genotypes vary differently. And then, well, once you have, if you are not able to observe these key traits, well, then you go on to some testing scheme. And actually, a lot of what we've been thinking about for the past while is how do you optimally design such testing schemes? So how can you come up with ways of finding out compatible cropping partners and good genotypes for these combinations? And that's something I'm going to look at again in a little bit of detail. And then the final quadrant here. Well, I'm just going to go through these and I don't need to make any sort of preparatory comments. So that's a bit where I'm going to go through. So I'm going to go through these one by one. I give you these exciting red stars to show where we are on this wheel. And we're going to start with assessing variation. Of course, again, I don't know what sort of background you have, but breeders, of course, are always looking for genetic variation because it forms the base of selection. I mean, you can have variation in the field, but if you don't know the genetics behind that variation, you're just selecting on noise. So you want to be able to estimate, in a sense, genetic differences, and yeah, that's what forms the basis of most selection programs. Of course, for a breeder also would like to know beforehand how much variation, what are we talking about? What's the size of that variation? And we're talking essentially about variation components. So how big is the amount of variation for this genetic variation versus sort of environmental variation? And what are we talking about actually when I mean, in the previous presentation, you used performance, and for you, the performance was, as far as I understood, the sum of the wheat yield and the faba yield just put together in a bag and weighed. another breeder might say, well, I want the faba. I mean, it's much more valuable maybe than the wheat. So therefore, that maybe is something that you would like the system to deliver more. But even broader than that, I think we could probably think together of other performance indicators than just kilograms of grain. What else could we be thinking about? Taking maybe faba and wheat, seeing as it's been already talked about this morning.
Biomass.
Biomass could be exactly, if it could be a performance indicator.
Like fertilizer.
You could indeed. So you could start to couple the output with the amount of inputs that were needed to create that output. So that could be a slightly more nuanced performance.
Synthetic grid?
Say again?
Photosynthetic grid?
Yes. If a farmer, for instance, yeah, indeed, if you ultimately yield is often what farmers want to produce, and if that photosynthetic rate was highly correlated with it and was easy to measure, maybe it would be a good proxy trait for yield. There was biodiversity, exactly. So we could start thinking wider of just getting the cash from the crop. So indeed, biodiversity effects.
Effect on the soil.
Yes, biodiversity exists in the soil as well. So what's the contribution of these genotypes to the soil, fertility, so the organic matter level, the soil life, the microbiome.
But because now all the graphs show yield.
Yes.
If you do that, you have to show something else, right?
Yeah.
Like this land equivalent ratio, assuming the yield stays the same. I think that would be a really good idea because the yield is already so high, it's so hard to make the yield higher.
Yeah.
But you could try to make it more efficient in certain terms like biodiversity and less pesticide use, less.
Yes, I think it's crucial. Again, this thing, if we can maintain these yields, we don't have to continually improve them. We maintain them but at the same time reduce the inputs, and particularly the use of pesticides, herbicides, those sorts of inputs that are quite toxic for the environment, and also artificial fertilizers. But I do think, indeed, in a lot of these papers and studies, we keep focusing on yields. It's a very easy thing to measure, and it's quite simple. If we start adding what's the effect on insects, bird life, soil microbiome, and is it realistic for breeders to take those other indicators into their breeding equations? That's we're allowed to envision a future where they do, particularly if it's valorized. But just to raise this, I mean, often you'll see things about yield, and it's just because it's a good proxy for something that a breeder can target. But often one thing that didn't come up is, for instance, grain quality and bacon quality, protein content. These sorts of things are oftentimes also measured by breeders.
Yes, carbon sequestration.
Yes, yeah, but breeding probably isn't looking at that yet. But they're certainly looking at things like post-harvest quality, bacon quality, cooking quality, chipping if it's potatoes, and so on. So there's many things. But just to say, I might mention yield, but please don't think I'm just got a yield hat on. And there are a much broader set of traits that breeders could be trying to maximize and to optimize. But to get back to these ideas about these variances, we need a framework to estimate these components. And unfortunately, I know Vodka is here, and he said, "Please don't just give me statistics." So this is a little bit of statistics.
I love statistics.
No, of course I do. Of course I do. As long as they're linked in some way to reality. But it's funny. So this framework of estimating these variances was sort of, I would say, almost independently developed between plant breeders and also animal breeders. And of course, it's nice to think about animal breeders trying to breed friendly pigs that don't bite each other, or chickens that don't peck each other. And there is a heritable genetic component for social behavior in animals. Believe it or not, a breeder can actually select, or an animal breeder can select pigs that don't hurt or don't attack or bite or so on, don't have unfriendly behaviors. So these things were developed both in animal and plant breeding contexts. Now in plants, as I said, breeders are often interested in these genetic effects. And usually, if you've done some basic plant breeding course, you often think about the phenotype is made up of the genotype and the environment. This is sort of this classical breakdown of variances. But in fact, this G is essentially what we're talking about here. It's the effect of the whole genome of this individual on its own performance. And again, performance can be broad, but here let's just talk about biomass because it's simple. When we start mixing species, and particularly if these species interact, well then we may see some changes in those phenotypes. So we may start to see, for instance, in this companion plant that now it's put on some extra leaves because, well, it likes what this plant is doing with exudates in the soil and making maybe more phosphorus available to this plant to take up. And vice versa, this plant has also caused our original species here to get bigger, taller, more leaves, more root proliferation. So part of the phenotype of this plant here is actually expressed here, but it's a two-way process. So we've got these so-called direct and indirect genetic effects present in both of these species. And if we have a framework like this is just a basic description of an interaction. If we're able to measure differences, well, then we're able to assign effects to these things. Now, ultimately, when we come to trying to optimize performance, we're talking now about system performance. We're not just trying to optimize this guy because maybe the best genotype of this one might end up completely outcompeting and overwhelming this partner. So we would like to try to find some sort of way where both species essentially benefit. And I come back to this in a while about how we might do that and turn that into an actual target for breeding. But it's, well, there are ways to do it, shall we say. But part of the issue, as I said again, is that we're now trying to maximize something that's not independent between these two crops. These direct and indirect effects are measured in different crops. So again, part of the phenotype of this is now expressed in the partner crop. And that's an important thing to bear in mind. Now, there have been some studies on estimating the size of these variances. So I said a breeder wouldn't bother doing this if these variances were very small, these indirect genetic variances. There was an absolutely wonderful study came out last year from the work of Benedict. Now, Benedict's going to talk about this after the break. So, but just to say, it was quite interesting, particularly if you look at the fraction of yields. So these are mixtures between pea and barley. And in the barley fraction yield, just how large the indirect genetic effect of the pea was in that yield. So the identity of the pea in those mixtures had a big impact in terms of variation in those barley performance. So a breeder would be crazy to completely ignore this pea genotype effect here. Now, maybe of course, they'll find the best one and that's it and fix it, but we'll come back to that in a little while. That's one example in a system with mixtures of pea and barley. In Arabidopsis, there was also a really nice study just came out last year. They found evidence for these indirect genetic effects for biomass. You could say, well, the effects were rather small. In fact, the heritability here was rather small. So an awful lot of the phenotypic variance for biomass turned out to be non-genetic. But of the genetic component, about 10 or 12% turned out to be dictated by the identity of the neighbor. So I would say substantial, given the total amount of genetic variance for biomass there. Okay, so the first step, I guess, would be a breeder would do some sort of a trials, and that's what we've been trying to do the past number of years in a maize and faba system, and look for these direct and indirect genetic effects, just estimate these components. But we now have all these modern varieties. So we would also think, well, are these modern varieties suitable for the system, or do we need to go back to sort of land races and wild relatives? And I think wheat is a very interesting example. In your study, you had various different heights of wheats, but of course, a lot of the modern varieties of wheats for monocultures are these semi-dwarf or dwarf wheat varieties, also in rice. I presume some of you have heard of the Green Revolution. So wheat has been completely changed from how it used to be pre-Green Revolution. And do people know why these small stature wheats are more?
It produces less straw,
Yes.
and so we have more yield, grain yield.
So the harvest index is higher. Indeed, there's less effort being put into producing straw, and more of that those assimilates go into the. That's true. Harvesting is
machines.
Yeah, well, indeed. So And what's the main advantage, you know, for well, maybe even not even mechanized harvesting, but there's a huge advantage to these short stature wheats as well. No lodging. And so lodging is when a plant falls over. And do you know why these old varieties lodge a lot?
First of all, they were tall.
They're very tall, yes. So we have lots of winds now, whereas in the
Yes.
past they didn't have any wind, so they didn't fall over. So why would they have? Because in the past lodging must have been an issue as well.
Something with the nutrient, right?
Yes.
Like,
So,
The density.
or maybe well, I think my understanding is it has a lot to do with nutrients.
because the Green Revolution also not only the height, it comes with the input you need
Yes.
to add with it.
Yeah.
The problem is when the height is short, it also the root length is also short, so they cannot take all the nutrients. So therefore, you also added extra fertilizer and pesticide, which comes together.
Okay. Yeah. I also I understand that if you give these older varieties higher levels of nutrition, they do tend to they become weaker as well.
Yes.
So it promotes maybe lodging. Yeah, so in a way, we've taken wheat from what it used to be, and it used to be tall as well because straw was a product that was needed for bedding, for roofing, for various different applications. Yeah, we've completely transformed it into the short stature. So this could be an example of one where, yeah, maybe these modern, very short wheat varieties. So that's why your research is very interesting in this. That indeed, maybe we do need to think back to a different idiotype of wheat if we're going to be mixing it. Okay. Now the next point is about these G times M interactions. I'll just put my, as I can tend to talk and talk. I'm not careful. So yeah, so Moore et al. Suggested that G times M interaction is a good thing to test. Now I'm not taking from that. Just to refresh your memories on G times M or G times E. Here's a sort of an example of we have two genotypes and we're looking at their performance between two systems. So you could think of maybe this is monoculture and this is intercropping. I'm not quite sure, but anyway, just two different management regimes. If there's no interaction, well, then we just see that we can predict the performance of these genotypes in the second system if we just know the additive effects of the environment and, of course, the genotype themselves. So we can pretty much predict going from one system to the next. If there's an interaction, it means that these differences between the genotypes are not constant. So they can either, in this case, be divergent, convergent, or in the worst case, a crossover, in which case the rank ordering changes. And for a breeder, of course, this is crucial. If there's these particularly crossover interactions, then it can have implications for which varieties tend to be in your selection environment the best, might end up in your target environment where the farmer will be growing it, not to be the best. And there was this study, and in fact, I think it was one of your co-authors, Demi, referred to Demi on your slide. So it's a small world, but he did this nice meta-analysis of various different intercropping studies where they compared monocropping and intercropping. And of the studies which reported interactions between the genotype and the cropping system, about 75% of these turned out to have a significant G times M interaction. So yeah, question mark for the breeder, is that an issue? Well, actually, there's another parameter or another way of looking at this, which is called selection efficiency. And this is where you, instead of calculating and testing for statistical interaction effect, you figure out, well, how many of the lines or the accessions, we'll call them lines, that were selected in an intercrop trial would also have been selected in monoculture, and compare that by subtracting the number expected by chance to coincide between the two systems. So it's a fairly simple formula. And just to visualize it a little bit, if we have a negative selection efficiency, and again, this is our selection system, assuming now that this is a monocropping trial, so we're trying our wheat, for instance, in just monoculture plots. The alternative system is we're growing our wheat with faba bean. In a negative context, you're doing worse than chance. So in this case, you select all these lines, and this is with a selection intensity of 1/3. So you take the top third of your lines and say, these are the ones I want to progress in my breeding system. And in this case, well, very few of them actually, fewer than you would expect by chance, ended up being in the top ranked third in your alternative system. Now, as we start to increase those selection efficiencies, you can see what happens. Those lines start to become more and more collinear. And at the higher selection efficiencies, particularly up at the perfect example, your top third, so your top ranking genotypes here correspond exactly with the top chosen, so the best performing lines in your alternative system, so in your intercrop. The thing that's a little bit concerning about this is that for a breeder, this of course is quite important. Breeder wants to know, okay, this is my monoculture, it's a very easy test system, but I come with exactly the same top varieties in my alternative system. So my selection is completely efficient. But there you can still see cross rank changes, rank differences here. So here you probably have, or maybe have a G times M interaction, but a breeder maybe doesn't really care about that as long as their selection efficiency is 100. And when there was a sort of an analysis done, a meta-analysis on these selection efficiencies, or meta-analysis, so back in the day, they used to be called meta-analysis. This is from the 90s. They found that on like most of these studies, selection efficiencies were not optimal. They were at most 80%. And in some cases, they even got down to these negative numbers that I saw earlier. But just to say that G times M may not be the most relevant criterion for a breeder. This thing about traits, so of course traits are important. We would like to identify these key traits. So usually breeders know about these key traits. So we've talked about potentially expanding the range of traits, but of course yield is usually up there. Disease resistance is of course extremely important. But again, these traits are often context dependent. So other traits that may be important for intercropping may not necessarily be obvious to a breeder per se. So they have to relearn essentially what is the trait space that they should be targeting. Now, there's a number of approaches, I suppose, that you could take to this if you don't know what these traits are a priori. One would be taking an agnostic approach. So agnostic being not having any particular belief one way or the other. And that would be, let's just record as many traits as possible. And that's a little bit what we've been doing we've been flying drones, we've been using the trait seeker. We're just recording a lot of, or we can extract a lot of traits potentially from these high throughput platforms. And then maybe we could then find, well, these traits can explain variation in the intercrop.
Can I ask
yeah, go ahead.
a question to explain this perhaps a little bit better?
Yeah.
You're talking about key traits, right? And then breeders usually know traits like yield.
Yeah.
So could you make a distinction, or is it necessary to make a distinction between the traits that we can get from whatever platform we're using and the targets that breeders may be interested in? Because you could say yield, it's a crop trait,
yeah,
but it's also the target that they want to aim for by selecting certain underlying traits, right? So what is should this distinction be made, or is it of the same coin?
I'm not sure if it's a helpful distinction, and I'll say why. Because I think there's two schools of thought in terms of selection theory. There's the one school of thought that says, okay, yield is a complex trait, hundreds, thousands of genes underlying yield. We cannot just select one or two genes. It just has to be some sort. But to make it more manageable, let's select for photosynthetic efficiency, leaf angle, rooting depth. Let's figure out all these other traits that all somehow contribute to yield, and let's select on those. And some breeders really believe this is the way to do it. You dissect a trait into subtraits. I'm also a bit more like, well, if you just select it for yield, you'll also get there. And that's the thing. I mean, I'm really about root phenotyping. I think it's really lovely to understand roots and so on. But I also know if roots ultimately, if they contribute to above ground performance, they'll have just been carried along anyway. You will get greater branching. You'll get different angles in your roots just because they're the ones that contribute to better cabbage head yield, for instance. So I'm a little bit, I could see the argument both ways, and particularly scientists and researchers love dissecting things and studying everything in detail and getting a handle on it. Now we understand it. But a breeder, if they just keep selecting for above ground yield, they'll get the other ones along there with them without actually knowing about them. The difficulty here, though, is that yeah, a breeder would really love to not have to do all these complicated combinatorial designs and having all these testing. A breeder would love to be able to select for intercropping in a monocrop, because then they could just, it's a very simple step over. So knowing these traits would be a great shortcut to it.
It's the same case for crop quality, because quality is not carried out on yield.
I'm not quite sure I understand your question.
Like protein content.
Yeah.
Plant can have a greater yield, but a lower protein.
Yes. Yeah, indeed, there are traits. In the end, often we know about a correlation or covariance between traits. So protein content and yields usually are negatively correlated. But if they're negatively correlated, but the correlation is not -1, so say it's -0.5, it still means you can improve both yield and protein content. Only if they're 100% negatively correlated that you're locked into it. But usually there's space to improve both despite trade-offs. Okay. So that one way, just to get back to this way of finding out what are the important traits, you could take an agnostic approach, record everything and see, try to uncover some that way, or you could take a more informed approach. So using, for instance, theory, what's known already from ecology, but interactions, complementarities, or you could use these crop growth models. And I'm maybe I better be careful because of who's sitting in the room. So there was a study done in around 2015, 2016, looking at intercropping of maize and wheat, a rather interesting combination of crops, I would imagine, but apparently it has some advantages.
It works very well in there's a tradition in China, but
Okay.
in real life.
The conclusions of that study, again, I have to be very careful because of who was co-authors in this paper.
I don't remember it.
Okay.
Don't worry.
It turned out that there was a yield advantage in the wheat because in the intercrop you got more reproductive tillers in comparison to the monocultures of the wheat, and you also had larger leaves in the border row plants, so there was actually plasticity as well, which was playing a role there. So that was very important for yield advantage. So this is again an application of where you use, of course, these were parameterized using experiments, but when you were able to verify or uncover and really get a handle on some of those traits and that were important for the for the performance of that intercrop. An alternative approach, and again, I better go through this slide quickly because this is what Benedict did. Suppose you have some ideas about key traits that are important, and you can test then, if you have measure these, test whether they're associated with your mixing effects. So GMA here being a general mixing ability, which is, well, it's related to some of these direct and indirect effects. And Benedict showed that very nicely. I even have a picture. This is from my back garden, Benedict. I was growing peas earlier this year, of a stipule, because I had to look up what is a stipule, but Benedict will talk more about it. But indeed, they found a correlation between stipule and the mixing ability of pea. But I don't want to give away stuff from Benedict's talk, so I'll leave that for there. If you have questions about it, you can ask the man sitting at the back. Okay. Now, Virginia also mentioned testing crops with many partners. How am I doing for time?
You're doing absolutely
okay.
fine.
Yeah, okay. And there are approaches, and in fact, you'll meet Dirk van Apeldoorn, who's one of the authors on this study, where they come up with a spatial compatibility score. So this is using ecological theory, using databases, their own trials, but also other trials, to come up with some score to figure out how well different species combine with themselves. And in this case, they've also overlaid it with the gross margin that a farmer can expect to gain. So you can identify good and bad combinations from this approach. The only caveat that I have is that these sorts of studies are still being done without a genotypic component. And what I mean by that is that they're being done with a single variety, usually, or at most two. But usually it's just single variety studies. And I put this out there. Of course, this screen is not very good, but you could imagine there's a sort of a search space that we have. Imagine we've got these two crops that we're figuring out, do these crops combine well? In a sort of a neutral crop combination, I imagine there's some variation, that some combinations will end up looking better or worse than each other. So in a good combination, yeah, you may be lucky to find one combination of genotypes that gives you the correct answer for these crop level interactions. But you could also just as well be unlucky because these things are of course, quite variable. So I imagine there is some sort of a continuum, a spectrum of potential conclusions you can draw on a crop basis that's dependent on the genotype you choose. Yeah.
Each square represented or a genotype?
This is a combination of two genotypes in some sort of a trial. Now, I just made these numbers up. So they're just fake numbers, random numbers. Yeah. But just to give an idea that we have to be a little bit careful about making a crop combination conclusion based on one of these squares. That's all I'm saying. But it could be that I'm wrong, that this is just all white, this is all blue, and blue meaning a favorable, and this is all pure red. But I don't think so.
If you do it in 2 years, you can publish it in field crop research. Yeah. One variety of wheat, you choose one variety of baba, it's totally okay.
Good to know.
The breeding journey.
You do that all the time.
So if we start introducing genotypes into this equation, we really open a Pandora's box. So suppose we have n crops, each of these crops has genotypes. If we do an unreplicated full factorial design, this is the amount of combinations we have. And just to visualize what that number looks like, suppose we're interested in assessing the compatibility of two crops. So that's this blue line down here. You'll see that if you increase the number of genotypes up to only 10, you're already, and this is a logarithmic scale, so we're already up at, well, I'm not sure what this number is, but hundreds or thousands indeed.
Yeah.
If, again, a fairly modest experiment. Suppose you've got 10 crops you're interested in. Again, we're not, there's hundreds of possible crops. These numbers, again, yeah, well, they become astronomical. And again, this is relatively modest, you would say, on paper. And this is unreplicated, of course. So if you want to replicate any of these things, it goes through the roof. Now, Benedict is shaking his head there because, as Benedict well knows, incomplete designs actually give you, in many cases, quite good estimates of these interaction effects without necessarily having to do absolutely everything. So, of course, the previous slide was a little bit jokey because you wouldn't do it. But there are smarter ways of doing it. So these incomplete designs are quite powerful. But anyway, just to say, there is good reason why these crop compatibility studies are usually done with a single genotype or at most two, because otherwise it just becomes unworkable. But I don't want to get your hopes down. Huh? We want to stay positive this morning. The final thing is this.
The whole week, preferably.
is the final point, and this is where I think we're getting really to the good stuff, is selecting for intercropping. So Moore et al. Suggested that, well, you've got three flavors. You've got select your crop separately in your monocultures. Again, this is again for breeder, really what they would like to do. Or you could go a step further. You could start to use a number of testers of the companion crop. So instead of testing all the combinations, you take one or two representative fabas, like you did, for instance. You took two faba testers and you tested them against your panel of wheats. Or you could do something a little bit more well, where you're not just biasing your system, shall we say, very much towards your wheat, but if you want to simultaneously improve these crops, and the conditions of your decision tree have got you to this point, well, then you go to a sort of a co-selection program. I'm going to look at those in turn. Well, of course, selecting separately in monoculture, it's the simplest approach. And it's very important to know this correlation between your intercrop and your monocrop performance, this row. Now, whether it's Spearman or Pearson, I'm not really that worried. But what I was interested to see is how does this row, I think in this case, it's a Pearson correlation coefficient. How does this row correspond to the selection efficiency that we saw earlier? And in fact, they're very linearly related, particularly when you're talking about positive correlations. They only start to go a little bit, start to take different trajectories if there's a negative correlation between monoculture and intercrop. Okay, so that would be just as useful an indicator as selection efficiency. Now, this row, this correlation between systems actually affects your genetic gain. And there was a nice study from Banchik et al. That looked at this. They looked at three different levels of this correlation. So from poorly correlated, again, which is around the number you found, and this was the highest correlation. You found 0.89. It's perfect that you gave the talk just before. So we have, this is actually in real life. You could find these range of correlations, and you can see here if they're more highly correlated, this was a breeding scheme that they simulated where they were selecting in monocultures and testing them again in simulations. They were testing them in intercrop between fictive, made-up species, this study. But anyway, with a high correlation, you can see you just get a much larger gain, and of course that makes sense. You've got much more fidelity between your monocrops and your inters. On the other hand, when it's lower, you see the gain is lower, but not only that. If you look at these lines, they're to do with different types of genomic selection schemes. I won't go into the details because I would have to refresh my memory on what all the different ones are, but they found the order here changed a bit. So the best strategy for a breeder actually changes depending on this correlation between the systems. So that was quite a nice little study. For the second type, and this use of testers. So suppose this correlation is quite low. It's no longer, you won't faithfully predict your good varieties by your monoculture testing scheme. Well then, using tester varieties, so using some representatives of the other species is a good way, I think, of starting to breed for intercropping. It's very analogous to the use of testers for combining ability estimation. For those of you who know about F1 hybrid breeding, oftentimes you use a couple of testers of the other. In this case, they're trying to combine parents to come up with a good F1 hybrid, but it's quite a similar procedure. You estimate first a general combining ability, and you use a couple of testers of the other the other parent to get an estimate of your combining ability, and then you can go for specific combining abilities. That tends to be what's done in hybrid breeding often. In mixtures, you could have the same thing. If we have these two crops, if we didn't have testers, you could imagine for replicates, you would have N1 times N2 plots. You can reduce that down to the sum of those if you have a good tester of the other crop. So it would just reduce your numbers quite a lot. So it's a bit of a compromise in a sense between monocrop breeding and simultaneous intercrop breeding. You have to be careful that your choice of tester line is, of course, very important. It has to be representative. And it's experimentally simple, but it may have some disadvantages, and you have 2 minutes to chat with your neighbor about this. So I've been doing a lot of talking. It's a moment for you to have a chat with the neighbor. What could be the disadvantages of this tester-based approach? Or you don't have to discuss with your neighbor. You can discuss plenarily. Or just think about it for yourself. There's the thing in educational theory, it's think, pair, and share, or something like that. Sounds modern. It sounds modern. Yeah.
I wouldn't know how to choose the tester.
You wouldn't?
No. Every variety.
But you make decisions. So in this previous study, Fanfare and Mallory were chosen as faba being, in a sense, testers. They were chosen maybe for their differences in their plant height or their yeah.
You can miss the best combinations, right? If you substitute the value, if you don't use all testers, you can perhaps miss optimal combinations.
We have to just, maybe we'll clarify this. Suppose we're working with wheat and faba, and one breeding company is a wheat breeding company, another breeding company is a faba breeding company. So the wheat breeding company takes a couple of the fabas, plants them and tests their wheat and develops their best wheat. And the faba breeding company does the same. So maybe think of that as the context. Because if you have a breeding company that has both, they have the option. But maybe some breeding companies won't want to be messing with the faba. They're just the wheat guys. So for them, it would be, indeed, they could miss some combinations. But we saw the numbers exploding if they include all the combinations. So choices do need to be made somewhere.
I guess the more you want to go agnostic, the more you don't know if you're doing it right.
Yeah.
Right. So if you are interested to know something about the mechanisms or have some hypothesis, then you can choose extremes of one condition like short and broad. But if you really don't know,
yeah,
you do it blind.
Yeah.
And then
I agree.
It's risky, I guess.
I agree. Very, very true. It could be that the wheat breeder says, well, Fanfare is number one on the market, or some Lima Grand varieties. If we're number one on the market, that's the one that most growers are. So they say, well, I don't really care about whether it's the best or compatible, but it's just a market-driven choice here. could also be. But I very much agree with you. If you don't know what are the traits and what are the compatibilities, particularly with, for instance, maturity class, sowing time, compatibilities with phenology, those sorts of things as well as the architecture, then picking one could be risky if there's a lot of variation in that other species. Yeah. Yeah, very good. I think that's our 2 minutes of discussion time. Not everybody said something. Okay. Now, and the final point then, it's the sort of the cherry on the cake, is it? This co-selecting crops simultaneously. So this is, again, we come back to this little drawing of our two crops. But what's important here, or what can give us a handle on this in terms of selection, is to introduce some way of combining these direct and indirect effects. Because of course, as I said, again, this direct effect you measured in this crop, but indirect effect is measured in the second crop. And it's a bit like selecting elephants and mice. You know, they think maybe these are pumpkins. Yeah, so we got kilos of pumpkins, and maybe this is wheat. So they could be completely scaled differently. We can't just sum up these components without doing something. So one way that you could think about is introducing economic weights. So if these two crops have different economic weights or values, sorry, again, this is a yield example, but if we know how much a kilo of grain, of wheat grain, and a kilo of faba beans, then we could introduce these two economic weights, and then we have something that's a target of selection for crop one, and vice versa for crop two. So we now have a handle on how to improve the system performance by selecting on these combined, these weighted sums of these direct and indirect effects. I guess this comes back to this idea of having good information about these indirect effects, and using a single tester approach, we're running the risk that we're essentially estimating only these indirect effects in a single genotype of the partner crop. So we're making a strong assumption that there's a very good correlation between these indirect effects as measured in the tester and the indirect genetic effects of these sort of true. And this true indirect is, I would imagine, some sort of an average across all possible combinations. So yeah, we're assuming that there's very little interaction between these direct and indirect effects. Speed up a little bit, I think. Yes, Benedict is saying speed up. No. Well, no, he's not. Okay. So if we are a company that has both wheat and faba, and I like using wheat and faba, there's a great example. Well, then maybe we have both of these. So maybe indeed we could randomly choose individuals from both crops to estimate these effects. And again, my colleague at the back has talked about designs that can be used and done some really nice work on designs that can be used to assemble those random combinations. This is a bit of an aside. I won't bother going into it, but that's just more for a modeling issue. Yeah, up to now I've been talking a little bit about variances, but also covariances are important. So covariance is how these two things vary with each other. Is there a correlation between these direct and indirect effects? You could imagine a negative correlation between direct and indirect effects for biomass. Go back one. Why would there be a negative correlation between indirect and direct effects for biomass? Or because they're competing, yeah. They compete. So if one plant is big, then its neighbor probably is. If there's a strong competition effect there, that the neighbor will lose out. And
but this is a negative correlation between the direct effect in one species and the indirect effect in the other, or the direct and indirect effect in the same species.
So this again, we'll call him John. John is our cabbage. Yeah. John has got a big biomass.
Yeah.
Whenever John is next to you, he's taking all the nutrients, he's shading you, he's taking all the water. So his indirect effect is actually a negative one. You're going to be less than the average when you're next to John. He's a big neighbor.
But the indirect effect of John is measured in feet.
Exactly.
Okay.
Or in the species of peats.
I like that. Okay.
So part of John's biomass and how does his neighbor look?
Yeah.
And unfortunately, when he's a big cabbage, the neighbors are not looking very happy next to him. Yeah, and so you can imagine a small neighbor, we'll call her Jenny. She's very friendly. She takes a very little space. She only nibbles a bit at the nitrogen in the soil, and all the neighbors nearby are having a great time. So they're above average. Okay, so that's for biomass, but there could also be other traits. Could we have a positive correlation between? Okay,
It's a specialization.
but there could be one thing here again is if we think about another trait like plant height. This could be a case where if plant height, you'll get a positive correlation, but that's more shade avoidance response. So you can imagine a taller plant, its neighbors then start to also grow.
It's one trait on the other trait.
Yeah, well, usually it could be, but it doesn't have to be in the same trait. You could divide these differently if you wanted. Anyway, just to say that there's complex relationships, but we need to know something about these correlations actually if we're going to breed for the system. So in Benedict's study, and I'm borrowing a lot of Benedict's slides, he's not happy about this. There was a negative correlation.
Indirect effect.
They found this was for yield, but indeed they found in both of the pea and the barley a negative correlation, and in the Arabidopsis study, pretty much a straight line. My gosh, what's going on with it? You can barely see the points, but anyway, a very strong negative correlation for biomass in Arabidopsis. Okay, and then the idea is, well, how do we come up with a breeding scheme? If we know these parameters, so we know the size of these variance components, we know something about the covariances, we know something about the economics, well, we can actually figure out for breeders what the best strategy is. And a number of studies have taken that approach. We had a good student last year who also looked at this in a faba bean maize context. And he looked at two scenarios, and this is where I'm going to end. The first was when the faba bean was twice as valuable as the maize. And the second situation was when the faba and maize were equally valuable. Okay, so we looked at two scenarios. We had for both crops a negative correlation between direct and indirect effects. Okay. In the case where the faba was the more valuable partner, what we found actually is that the system more or less promoted faba beans that had a high direct genetic effect. But in the maize, something interesting happened. You just saw that the maize essentially you got a lot of genetic gain. So on this axis is the sort of a genetic value, which is the sum of all the added genetic effects. I won't go into too much, but essentially this means that we got improvement in the maize for the indirect genetic effects. So the maize became a partner species, a companion species essentially for the faba. The maize that ended up being selected in this breeding scheme were more or less just a helper to maximize your faba yield. But this is over 20 generations of recurrent selection. If those weights were the same, so in this case now faba and maize are equally valuable, what happens in the system, you actually get an improvement of the direct genetic effect of both of the species. And in this case, the indirect effects also came up in a positive sense, which is, yeah, well, you could, although they're negatively correlated, still these indirect genetic effects are contributing to the performance of the partner species. So they also, interestingly enough, improved. But it just to show that what a breeder, if they knew something about these variances and covariances, they could figure out strategies and play around with different breeding program designs. And that's a little bit what we're doing at the moment in some of our work. Okay, so some final guiding principles, and I'll finish at this, and apologies for going over time, but I think we had the discussion within the presentation, so I'll get away with it. So if you've got a high correlation, if you've got a high correlation between the performance of monos and inters, so that's also what we're calling this high selection efficiency, you can very much make progress using monocrop. But this gives us no information on these indirect genetic effects, of course. I mean, you won't say anything about wheat's influence on faba if you don't grow it next to faba. The ratio of these effects, these indirect to direct, I think for a breeder is important. Because yeah, if this thing is very small, if these indirect effects are very small, why you could probably get away with ignoring it. So knowing the sizes of these components, I think, is essential. If the correlation between these indirect and direct effects is anywhere above -1, it's possible to improve the system to simultaneously select, like we're talking about these protein content and yield. You can still select your way towards improving both, but that has an effect or an influence on your rate of genetic gain. So how fast you make improvements over generations. Yeah, and we found, and we showed you there, these if you're doing an index selection based on economic weights, it's the relative weighting of the economic weight, shall we say, of these two crops that really defines how these indirect and direct effects evolve in the system. Okay, now we've been involved in a project for the past few years. We've been doing trials primarily with maize varieties from different breeding companies, but also faba bean, and also mixtures of quinoa. I didn't talk at all about our mixture experiments, but anyway, there's some references there.